what we want to be aware of is that on the donor, the dead S, the donor chromosomal DNA had genes for capsule. So I'll just put a little capsule here. So the donor chromosomal DNA had the genes for enzymes to make the capsule, right? Okay, so when the donor died and released the chromosomal DNA, okay, so here's donor chromosomal DNA. And what I'll do is, usually the circular chromosome, when the cell breaks open, the circular chromosome gets broken up into pieces, so you end up with little linear pieces. So we'll just show a linear piece of DNA carrying the capsule genes. The genes for the enzymes that permit a bacteria to make a capsule. And what we now know is that in nature, some bacteria have the genes to make DNA binding proteins. So let's cartoon some DNA binding proteins in here. We'll make them in orange. So it turns out Streptococcus pneumonia has genes for DNA binding proteins, and at a certain point during growth, they will make these DNA binding proteins, which will be located on the surface of the cytoplasmic membrane. So these orange dots are DNA binding proteins. Cells which are expressing, making DNA binding proteins are said to be competent. They're competent bacteria. And this is one of those circular arguments, circular definition. A competent bacterium is a bacterium that can take up naked DNA from its environment. Another way of saying that, a competent bacteria is a bacterium that can be transformed. Right. So in this process of natural transformation, this occurs in nature, the DNA binding proteins can bind naked DNA, and it's not that we're being risque here. Naked meaning the DNA is not enclosed in another cell, it's not enclosed in a virus, it's just naked DNA in the environment. The competent bacteria can bind to naked DNA in the environment, such as the donor DNA carrying the capsule genes, and the DNA binding proteins bind to that naked DNA, and then they pass it through the cytoplasmic membrane into the interior of the recipient. Okay, so we want to remember the living R string is our recipient. All right, now what do we know, you guys, about linear DNA? What's going to happen to it within a bacterium? Will it be destroyed? Yes, right? So how would the donor DNA carrying the genes for capsule production, how would those genes, that piece of DNA, how is it going to survive inside the bacterium? What must happen? You guys, yeah, homologous recombination, right? So it's possible that in the R strain, it may be that it was a mutant. It might be that at one time it had functional genes for capsule production, right? But maybe through a mutation, the genes either weren't expressed or the mutant proteins weren't capable of synthesizing the capsule, right? So we would have had homologous sequences on the recipient's chromosome. So remember, in homologous recombination, donated DNA can end up replacing homologous or similar sequences on the recipient's chromosome, right? So I know you guys get frustrated because I get to erase like that, but I'm going to do it. So through homologous recombination, that incredible crossing over event we talked about, the donor capsule genes would have replaced the mutant capsule genes of the recipient, and now the recipient has functional capsule genes. Meaning that those donor capsule genes, when transcribed and translated, would make functional enzymes to make what? Capsule, right? So now the transformed R cell can make the capsule, right? And there's no way we can tell it just looking at it. We can't tell, is it a transformed R cell or is it an S, original S cell? Does that make sense to you guys? Hopefully. It's just crazy these things are happening out there in nature. Okay, now with regard to cell and molecular biology and microbial genetics, it was heartbreaking to microbial geneticists when it was discovered that E. coli does not make DNA binding proteins. That means that in nature, E. coli can't become competent, and therefore our genetic workhorse in microbial genetics, E. coli, could be used in experiments in which it could be transformed with genes from other organisms, right? That was a heartbreak for the microbial geneticists. So what those folks discovered, there's a simple treatment that you can do in the lab to make E. coli artificially competent. And it's simply incubating the bacteria in cold calcium chloride. And then you transfer the DNA into those artificially competent E. coli in a process called heat shock. You quickly warm the E. coli up with the DNA that you want to introduce. By increasing temperature, the cells, the DNA start moving faster and faster. And when you get a collision at just the right angle, you can drive the DNA across the cell wall and cytoplasmic membrane of these E. coli that have been incubated in cold calcium chloride. So this will be a topic for next week in lab. And this making E. coli artificially competent and transforming them by heat shock is potential topics for both lecture exam 3 and also lab practical 2. In the D2L section, under the Chapter 8 module, there's a URL link to a video that describes and illustrates the transformation of E. coli using heat shock. So that would be a good little video to examine so that you'll have an understanding of how heat shock works in the lab to transform bacteria. Okay, good. All right, so transformation then, one possible definition, bacterial transformation, is the uptake of naked DNA from the environment by a competent bacterium. So on a short answer section, you guys, I might ask you to define transformation. And again, one possible definition would be bacterial transformation is the uptake of, fill in the blank, naked DNA from the environment by a blank bacterium, fill in the blank. Competent bacterium, excellent. You guys just got 100% on that one. Good, okay. So what we're going to do next is move to a second way that bacteria can transfer genes horizontally. And this is the process of bacterial conjugation. Now, as humans, when we're learning new information, we often take new information and we hang it on a hook of some topic we know already. So I want you to understand that many people inappropriately describe bacterial conjugation as bacterial sex because it's the closest thing to sex to us, right? But it's so important, folks, for us to remember that this is not true sex. Sexual reproduction is when two genetically distinct parents combine genetic information, and that recombined, that unique combination of genes becomes the genetic information for an offspring, a new cell, right? So remember, in horizontal gene transfer, this is not sexual reproduction. This is simply one donor bacterium transferring DNA to a recipient. But you will see people calling this bacterial sex, maybe for obvious reasons. I don't know why they call it sex. But okay, so let's talk about this. We'll use the model of good old E. coli, our workhorse of microbial genetics. And so we're going to talk about conjugation in gram-negative bacteria. And gram-negative bacteria are famous for making these incredible long sex pili or conjugation pili. In our model, they're also called F pili for fertility pili. In gram-positives, conjugation is not so dramatic. Gram-positive bacteria don't make these long, beautiful sex pili. Proteins are involved in conjugation, but it's a much more subtle process. It does involve cell-to-cell contact, but we'll just have surface proteins that bind the donor and the recipient together. So gram-positive conjugation, it isn't as glamorous. So we're going to focus on the gram-negative conjugation, the first that was discovered. So a little bit of background information here. And again, this is the second type of horizontal gene transfer. So number one that we discussed was transformation. What's this called, you guys? What's this called? What we're going to talk about? Conjugation, good. And conjugation the donor is alive, right? Okay, in transformation, the donor was dead. And in the third example, horizontal gene transfer transduction, we'll also see the donor is dead. So this is the only time where we see a living donor. All right, so again, we're going to use the historical model of our example will be E. coli. And for an E. coli to be a donor in conjugation, the E. coli has to carry a special DNA sequence. A special DNA sequence called the fertility factor. The fertility factor. And as biologists, we're so bad, you guys, what do? We just use abbreviations. So you often see this simply as the F factor. Now remember the F factor, it's a special DNA sequence. It encodes the genes for formation of the sex pilus, and it also encodes genes that permits the donor bacterium to transfer DNA to the recipient. So another interesting tidbit about the F factor is it can exist independently of the chromosome in a plasmid form, in which case we call it the F plasmid. Or what's really cool, it can insert itself into the bacterial chromosome. So we'll look at both of these situations. In both cases, the donor bacterium carrying the F factor can act as the donor in conjugation. So what we're going to do is we're going to take a look at a couple of matings. And again, our terminology is really inappropriate because mating suggests sexual reproduction, right? So some of our terminology is a little bit misleading. But let's take a look at a couple of E. coli conjugation matings, and we'll put it in parentheses. And what we'll do is we'll put our donors here, our donor, and then we'll put in X to say this is going to indicate that this is our donor that's going to conjugate with a specific recipient. And the first mating we're going to look at is an F plus donor that's going to conjugate with an F negative recipient. And again, folks, sometimes as biologists we slip up. Often we'll call the F plus donor the male, and we'll call the F negative recipient the female. So just be aware that sometimes this misleading terminology slips in. Okay, so the cool thing in nature, one might ask, say in your intestine, how would an F plus donor know that there is a recipient in the neighborhood? Well, bacteria use chemical messengers called pheromones to communicate with one another. And these chemical messengers, these pheromones, permit donors and recipients to become aware that a mate, an appropriate mate, is in the neighborhood. Even advanced animals like ourselves, we too use pheromones. Yeah, they're important chemical messengers usually involved in reproduction. So when a donor detects that a recipient is in the neighborhood, the donor starts to synthesize the proteins, the little protein subunits that are assembled into a hollow tube, and the hollow tube is the sex pilus or conjugation pilus or F pilus. The recipient, in the meantime, has synthesized unique proteins on the surface that will act as a receptor for the tip of the pilus. The whole purpose here is that the donor wants to make physical contact with the recipient by using the sex pilus, and then once contact is made, the really cool thing, the donor then disassembles the protein subunits of the F pilus. That causes the F pilus to shorten, and it will bring the recipient into close physical contact with the donor. So we end up at the end of that process. I mean, these could be gram-negative bacteria now in the process of coming into close physical contact. And it's when they're in close physical contact that the donor will transfer a copy of DNA to the recipient. Okay. So now let's look at some specific examples. We'll first take a look at an F plus donor conjugating with an F negative recipient. And just to get you oriented here, you guys, here's our F plus cell, this is our donor. And here we have the F factor as a plasmid. So I would have called this the F plasmid. Here's the donor's chromosome. Here's our recipient. And notice that the recipient is F negative. This is the recipient's chromosome. So this is following shortening of the sex pilus. So at the base of the sex pilus, there's a protein apparatus that functions like a hypodermic syringe and needle. And it's called a secretory system. It's through the secretory system that the donor passes a copy of the F plasmid to the recipient. Okay, so here we see a copy of the F plasmid passing to the recipient. So at the end of conjugation, the donor remains donor, F positive, but what's happened to the recipient? The recipient is now a donor, right? Because remember, donorability is based on having the F factor, right? So now the recipient has the F factor. So the recipient is no longer a recipient, the recipient's a donor. And yeah, I was sharing with your colleagues yesterday, they did think it was funny, I don't blame them, but I was saying, biologists, we spend all the time in the lab, don't have this, wild social life. So when we have parties, our jokes are pretty nerdy, right? So one of the jokes at a biology party is we talk about conjugation and how sex is contagious, right? Because in this case, female is converted to male, so it's, yeah, your colleagues yesterday didn't think that was funny either. Okay, all right, I'll just have to skip that joke. Okay, a good question that's come up before is that one of your colleagues said, well, what good is that, right? Why did this process evolve? Why was it maintained? And it's a great question. And I would suggest it's not the F plus F negative mating that provides genetic diversity to bacterial populations. It's the next mating. All right, so in the next mating, our donor is going to be a little bit different. The donor is what we call an HFR cell for high frequency of recombination. Now, our HFR donor has the F factor. But in an HFR donor, the F factor has inserted into the donor's chromosome. So here, we're seeing conversion of an F plus donor into an HFR donor. So we start out with the F plasmid, and there's a specific site on the chromosome to which the F factor will insert itself. So it's not exactly a homologous recombination because the donor maintains all the chromosome. It doesn't lose any chromosome, but the F factor is going to insert itself into the chromosome. So here we see this the F plasmid preparing to insert, and here's the F factor inserted or integrated into the donor's chromosome. So now we have an HFR cell. So an exam question, you guys, would be, where is the F factor in an HFR donor? Integrated into the chromosome. Good, excellent, all right. And it's these HFR donors, when they conjugate, we're going to see the HFR donors are going to transfer copies of their chromosome to the recipient. And I think that's why conjugation has survival advantage, because we're going to get transfer of donor genes to recipient, and that's going to increase genetic diversity in the population of bacteria. That's why I think the process has survived. So let's take a look then at an HFR conjugating with an F negative recipient. So our recipient's the same. So here's our HFR donor, here's our F negative recipient. And what's intriguing about this process, you guys, is when the transfer begins about at the midpoint of the F factor. And again, it's a copy of the F factor, a copy of the donor chromosome that will be transferred. So transfer begins about at the halfway point of the F factor. So if we can kind of imagine that, the first half of the F factor is transferred, and then it drags a copy of the donor's chromosome into the recipient, and it's only at the very end does the second part of the F factor get transferred to the recipient. Now, this process in nature usually takes about 90 minutes, an hour and a half. And you can imagine if this was occurring in your intestinal tract with the passage of food and feces and water, this delicate conjugation bridge is usually broken before we have full transfer of both pieces of the F factor and the entire donor's chromosome. So usually these end up being interrupted matings. So we'll get the first part of the F factor transferred, and then just part of a copy of the donor's chromosome. So as a result, usually in HFR matings, usually the recipients remain F negative. They have to have both parts of the F factor to be true donors, right? So in interrupted matings, usually the recipient remains F negative, but what has, she acquired? She has acquired some donor chromosomal DNA. And again, this chromosomal DNA would have to insert itself by homologous recombination. So in this way, in these HFR matings, the donor could transfer antibiotic resistance genes to the recipient, could transfer toxin genes, could perhaps transfer genes for capsule production or other virulence factors. So again, I think it's the HFR matings that provide genetic diversity to the bacterial populations and thus increase survival of the population of bacteria. Good, all right, so, good.
Can you repeat that one more time where you said that the HFR is found in an integrated, I need to get the, I was on the last slide right before.
Sure. Okay, how about if I do it as practice questions? Does that work, you guys? Okay, so in an HFR donor, where is the F factor? Integrated or inserted into the chromosome. Okay, right. In nature, and an HFR conjugating with an F negative recipient, in nature, does the recipient, does she acquire the full F factor? Usually not, right? So in an HFR mating, does the recipient become F positive donor? No, usually in nature not. Right? Does that help you guys? Just like, because I know there's all this new vocabulary and you're trying to get the words down and it doesn't give you time to kind of visualize what's going on. Yeah.
So since it doesn't acquire or become F positive, could it recombine with, say, another?
It could. Yeah. Excellent, right? Because the recipient remains a recipient. She can conjugate with another HFR donor that maybe has a different version allele of genes. And so conjugating F negative recipients, conjugating with HFR donors, they can acquire lots and lots of new genetic information and still remain recipients. So it's a fantastic way to acquire new genes. Yeah, and you guys, with bacteria, we're talking about survival of the population. We're talking about survival of the population. So anything that increases genetic diversity in the population increases the chance that that population will survive better in a changing environment. Yeah. Was there another question you guys were commenting? We okay? Okay, we're going to keep going then. And the third example of horizontal gene transfer is called transduction. And we're going to do transduction really light, you guys. And the reason is, to really get into the nitty-gritty of transduction, we need to know two different replication cycles of bacterial viruses, and we aren't going to get into those details until chapter 13. So we're going to do transduction light, right? And then in chapter 13, we'll come back to transduction and talk about it in a little bit more detail. Okay, so in transduction, a simple description of transduction is transfer of donor bacterial DNA to a recipient bacterium by a bacterial virus. So here, viruses are going to act as delivery trucks of donor DNA to a recipient. And again, just like in transformation, you guys, the donor bacterium is dead. The donor bacterium is dead, but the viruses will transfer donor DNA to the recipient. So what we're going to do is we're going to look at the simplest type of transduction, which is called generalized transduction. Generalized transduction. And generalized meaning that any donor gene can be transferred to the recipient. And this is the simplest type of transduction. And it is wild. It is crazy. Okay, so let's take a look at, and I know this is really tiny, you guys. This is figure 8.28 from your textbook. So let's see. Hopefully you guys can see this. So here's our donor bacterium. It's so nice when they have different colored chromosomes. So the donor chromosomal DNA, dark purple. And this little guy here that looks almost like a lunar lander, this is a virus that infects bacteria. And we call these bacterial viruses, historically, they were described as bacteria eaters because they can destroy bacteria, almost looks like they're eating them. So bacterial virus, meaning a virus that can infect bacteria. Historically, they were called bacterio. What's a scientific term for eating? Yeah, phagocytosis. So these bacterial viruses historically were called bacteriophage or bacteria eaters because many of them will destroy the bacteria, and it looked to early microbiologists like the bacteria were being eaten. So the bad news is, you guys, I'll flip back and forth. I'll say bacterial virus and then I'll say bacteriophage. So the important thing is to realize that they're the same thing. And often biologists will simply say phage. So the convention is, if I say phage, your brain is saying, oh, a virus of bacteria. So in this cartoon, our bacterial viruses, or bacteriophage, they have protein coats. So turquoise is the virus protein coat called a capsid. And one important job is that it protects the viral genetic information. So in this example, we have viral DNA. This is going to be a DNA virus. It uses DNA as genetic information. So in chapter 13, we'll see in the first step, the virus attaches to the surface of the bacterium. The second step is the virus injects the bacterium with its viral DNA. It's just the craziest process. And then once the viral DNA gets inside the bacterium, that bacterium is a goner. It's going to die. As Dr. Meyer describes it, once the viral DNA gets inside the bacterium, that bacterium becomes a viral producing factory. The bacterium is no longer going to make organic molecules for itself. The bacterium, all it's going to do now is make copies of the viral DNA and make viral proteins. As part of this process, the bacterial chromosome is hydrolyzed because it will act as a source of nucleotides to make more viral DNA. These viruses are the perfect parasites. And we just want to note, you guys, that often there'll be little fragments of chromosomal DNA in the cytoplasm. So after viral proteins are produced, the proteins self-assemble into these little protein coats, these capsids, and then DNA from the cytoplasm is packaged into these protein coats. And most of the DNA that's going to be present in the cytoplasm, most of it would be viral. So most of the little protein coats get packaged with viral DNA. But occasionally a mistake is made. And a little piece of donor chromosomal DNA will get packaged into one of the viral protein coats. So when assembly is finished, the bacterium will lyse, it will break open, releasing all the viruses, including the virus carrying donor chromosomal DNA. Now, as long as that protein coat is normal, this little, we could call it like a recombinant little virus, can still bind to a neighboring bacterium and inject the neighboring bacterium, in this case with what? Donor chromosomal DNA, right? So you see, you guys, how the virus is acting as a little delivery truck? The virus carries the donor DNA to the recipient and then injects the recipient with the donor DNA. Now, this will be